many people will teach you. I'm not the only one, but you'll get a little bit of a background, a foundational thing. Okay. So you already studied pathophysiology. So I assume that you have a little bit of knowledge as to what is a cancer, what is a carcinoma, what is a benign tumor, what is a malignant tumor. We talk about metastases, secondaries, and so on and so forth. So this is like basic information. Now if you, which one is the best? Black one? This one? Okay. Now you can imagine that in the context of immune system, the purpose of immune system is to differentiate between self versus non-self, right? Now cancer is a non-self. Okay, so immune system should identify and kill it. But at the same time, keep that in mind, the cancer of human cells. So they're your own cells which become cancerous as compared to microbes which are not your own cells. Does that make sense? So that's a challenge. So immune system has to kind of make a good identification. Now, like typical cycle, there is a cell, and it has it's just like in the modern marketing or shops, every product which is there has to have a barcode, has to have an identification number. Okay, and they're going to have a reader as well. I can't imagine in current day and time, so exactly a cell has that. So there are cells, they identify. And if you remember, it gives you one of the pictures where you have a antigen presenting cell, APC, right? And then you have a T cell. So this is like the marriage. This is something that, that has to happen at all times. So you have APC and we have a T cell, and they need to talk to each other. This is a communication. And remember, I gave you an example for the bar. If you have your cell phone, you see a lot of bar. So all these bar depends upon the communication. Okay, and tons of them. Now remember, we already talked about them. The major one is that if antigen is presented, this one, right? This is like an antigen. And it is presented by the antigen presenting cell in context with MHC class 1 or 2. And this is what happens. And we already discussed that. It's pretty much a repeat of what we already discussed. We need signal 1 and signal 2. Okay? Now keep that in mind. The cancer cell has to be identified, and one of the, it's just like, I'm not gonna do analysis for it, I'm just gonna ask you to give me your SS number for identification, or your class, university ID, and then everything should be there. So that is what it is. So the cancer cell or tumor has to be broken up by the antigen presenting cell, and just take a tiny portion of the sequence, which I just gave an example, like a typical identification, I'll say SS number, or whatever you want to call it, and present it to this one. So this is a normal system we have. And at the same time, we have tons of co-receptors. You see, we have tons of co-receptor which will determine, what needs to be done. It's just like building a house or a structure. So you can see a complete structure, but there is all the way from foundation, and actually I saw the other day, in front of my home as well. So firstly, there were some people who came, foundation were laid, and then, they came to do the plumbing, then came the electrician. So each and every company is a distribution of stuff. They're not going to come at one time, right? They have planned everything, so it's step by step, and then there is a management, there is oral. So exactly, it's just like a car manufacturer, where is all these things are laid out by even robotics. So that's what immune system does. So it has this reaction that comes over here once it identifies that, right? And then you can see we already talked about You know, some of them, for example, this one over here was for the cytokine, right? And then there are other receptors which are important, you know, in case they're all co-stimulatory. So they want to find out what needs to be done at appropriate time so that it becomes. Okay? Give another example that, you know, if you just pick up a person randomly, you know, and then there is a police and there is a, you know, FBI and there is CIA and, you know, all these things are complicated if there is some issue over there. Things are not kind of taken over by one. So exactly, you have tons of these receptors which are going to determine what needs to be done. Okay? And then response will be, but keep that in mind that this is done in good faith by the body, right? And the body does it naturally. So how complicated is that the cancer is picked up by the body naturally and dealt away naturally? We don't know. We are just learning, right? And as I said, our role as a pharmacist or health-related field is that we can find out if there is any deficiency in the system, right? So what we can find out is we can either have a positive effect, like you can see from here. We want to increase whatever effect is, or we're going to go a negative effect. So this is going to kind of balance out. It's just like giving an example of human physiology equilibrium, the best response. That's what it is. So is it up or down? It should be positive or negative. Body does it unknown. It's difficult for us, but we try, right? And that's what all these markers over here will have a drug, whether they're going to stimulate them or they're going to inhibit them, over here as well. So we have receptors, they're all proteins, and you can know, we can pretty much, it's like keys. So you can see all of them have different configuration. You see that? All of them have different configuration. So this means very tightly controlled. It's just like if I want to open something, so I have like 100 keys to open. Or you can give example that it's just like a barcode, and barcode with, you know, even for your ISBN number for the book, they are 13 or 15, right? So barcodes, they have a lot of numbers. So you can see this is what it is. If you represent that as a barcode, it's going to show as all these lines which computer reads. Right? And anything can go wrong in the cancer, all the way from identification till the end. Okay, so which number was that? 18%, 18.4. Where is 18.5? Okay, let's go there. This is an example of one of the things. Where is the title?
It's up there in the corner. I couldn't fit all of it.
Okay.
I just get the first sentence about the.
Okay, the journal schema for the adopted transfer natural occurring autologous tumor inflating lymphocytes. I think you will have a difficulty in understanding experiment. Let's pick up something else and then we'll come back to it, because this is all. So you can see that in the tumors, this is like, first one, when tumors arise in a tissue, a number of immune cells recognize them. So this is what we anticipate, right? So there is because the tumors are of organs. Organs are cells, right? And these are like cells. So all of a sudden, this is a typical cell, right. All of a sudden, something happens to these two cells, right? So they basically, if you remember your pathophysiology, they become abnormal, right? So we have tons of organs within our body. Anything can become abnormal at any given time. Okay? There's nothing you can do. And it's too small a change that may occur, but it does occur within our body, right? And what happens is, the very first thing that will happen is the immune cells will recognize that and eliminate them on their own. Now the question is, what type of immune cells are there. So you can see CD4 cells, CD8 cells, these are gamma delta, there's another, and then we have NK cell. So these cells will come and kind of bind to them, right? It's like an assembly line. If something goes wrong, somebody has to fix it. So that's the first step that is happening and should happen at all time within our body, without us knowing it. Does that make sense? Okay. Now what happens is, then we find out that this cell which is abnormal, right? Which is abnormal, and so abnormal cell is not going to function because it's abnormal, right? And the abnormal cell may kind of lead to other problems because it is not normal cell. Okay, so things are going in a different tangent. So what happens is that these cells are designed to identify them, eliminate them if they're working well without us knowing. Hopefully they are there. It's just like assembly line a product, there are some inspectors, whatever they are, they say no, go back, you know. So they are doing while the assembly is taking place. It's not a recall, like the car has been manufactured and no, it's not a recall. It's an assembly line process. The second thing is that the tumor, which is an abnormal growth, right? It's an abnormal and it's going to grow, right? And it's going to go out of control. That's what you learned in test 1. It's going to be abnormal, it's going to grow, it's going to be out of control, right? And it's going to become big in size, right? Tumor, or it's going to be either benign or malignant. It's going to even just be there, and malignant is going to spread. So you, at that time, you should know what is the difference between benign and malignant, what is metastasis, what are secondaries, and so on and so forth. All this test, probably DAS1 or pathophysiology 1, whatever that is. Now, the second chance is that there's a change over here. So this one now is a variant, right? So variant of, they are alike, but this is a different variant. So what happens is when there's different variants, 2 of the cells go away because they actually cannot recognize that. They can recognize this one, but it so occurred that variant tumor cells, they are more resistant to being killed because the more variancy is there, right, the more problems will be there. So that's a rule of the cancer, that if there is, because the variancy is very difficult to lead to. One of the things that will happen is that these 2 cells, for example, they were the one who were about to kill this one, but if it becomes variant, these cells will be ineffective. So what happens? You lose 2 cells. Okay? Over time, another variant comes. Over time, variety of variants comes. So another variant come over here, this one, right? So now what will happen is that this cell over here, or this cell over here, it also disappeared. It also, another cell leaves as well. So we had 4 cells, 1 variation, 2 gone, another variation, 3rd gone as well. And then we end up with a situation that we got tons of variations over here, and there is no cells to kind of come and kill them. So one variant will escape the killing mechanism, right? And then disappear. Remember I told you last week or the week before last week, the Nobel Prize was given to a guy who discovered the cells working 3. So he thought in his mind that who is going to regulate that? Is this like assembly line, or I gave you the construction firm, somebody's regulating it on the bigger picture, right? Overall supervisor in charge. So he found out that these are Tregs. So Tregs is a cell which is CD4 T cell, and it regulates. It's called Tregs, right? They regulate. But what happens in most of the cancer is there's still a mystery. We haven't found out that the cancer recruits T cells to protect it? That's a big question that we have. So, I mean, this is because cancers are weird. Cancers are dangerous, they recruit that. So once you recruit that, you are done. There's nothing. So you are now, happening is that cancer is growing uncontrolled. Any cancer, and it's gonna kill you. This is like a bigger picture of how the cancer mechanisms are. Okay? Now, keep that in mind, and if you come over here, on this one, where's the title?
Which one for 1806?
Where is it?
It's CTLA4 and PD inhibition.
CTLA4 PD inhibition.
Yeah, I couldn't fit it all.
Okay, so the drug which are available, like a couple of years ago, there was PD-1 blocker that was marketed for anti-cancer drug. So it's there. So one of the drugs which is there, we are using it, is a PD-1 inhibitor, or it is available in the market, and this is the current therapy of most of the cancers that we have, you know. And in this case, what happens is that, again, number one step will be we need cells to activate. So we have an antigen presenting cell that presents the antigen of the tumor to the T cell, and T cell get activated, right? We already know this story, T cell and MSC1, right? The other one we found out that these are the co-stimulators, right? These one too. And I told you that these are the ones that are required to kick in the activation of cell so that they can do what they need to do, right? In this case, what happens is that if, for example, this was a accelerator positive, if the cell, instead of having accelerator, puts on the brake, which is CTLA4, right? So what will happen is that this cancer is not going to be seen by the T cell, because it has to see it with a co-stimulator, right? So we call it exhausted T cell. So this means the T cell is just going to keep on pressing on and off, and it's going to kick in, because it's just like you have two prongs of this wire over here, and you're just pressing it, you know, and only one goes in, other goes. So that's going to work. So you have to have that. The third step is that in terms of treatment, right, what we found out that since it becomes exhausted, we want to treat activated T cell, right, so that it becomes strong activator. So what we do is that we want this guy, which is a negative not killing cell, we want to block that with an antibody. We block that with an antibody. So when you block that with an antibody, the negative will not come, so positive will come, and then it gets activated. It gets kicked in. So your T cell, which was there to kill the cancer, should have done this pathway of positive activate, but what happens is it shows off a negative breaks over here. So instead of CD28 binding with this, you have CTLA binding with this, so you have negative reaction. How do we treat that? We bind the CTLA4 with an anti-CTLA antibody so that it can kick in. So we use anti-CTLA4 antibody as a treatment. So that's one way of the treatment. Yes, no? So in the other case, you can see, for example, this is like a normal pathway. If there's a tumor cell, and tumor cell and antigen, and it presents to a effector cell, which is a T cell, right? So T cell, of course, has to look at this tumor cell with a co-stimulator. What we normally see is that for some good or bad reason, is that this cancer cell has an ability, the tumor cell, to go for a receptor which is called PD-1, and the ligand is PD-L. So L for ligand and the PD-1 receptor. So this receptor is on the T cell, and this receptor is on the tumor cell, PD1, PDL. Okay? So once it gets activated, this T cell is not going to kill the cancer, that we found out. Because cancer is weird. Cancer is going to act in a way that is going to escape our immune system. So these are like tons of experiments that have been done over ages. So this PD stands for programmed death. Programmed death. So this is like programmed death, and this is the receptor, major histocompatibility complex. We already discussed that. Okay. So what needs to be done now? So what we do is that since we find out this programmed death 1 is not working, right? So we want to block this one with an antibody, anti-PD1 antibody. We can block this one or block this one with it, so that this mechanism goes up. So we want T cells to see cancer antigen so they can kill it. The cancer is smart enough to occupy 2 receptors which have been, this is what the guy who got Nobel Prize 5-6 years ago, he discovered PD1 receptor, because this is what he thinks was the most important one, which was holding the cancer survive. So he came up with an idea, if you either block this one or this one so that this interaction doesn't take place, either with, an antibody for this or antibody for this, then the tumor cell will be killed by the treated T cell. So these are called, if you look at anti-cancer therapy, they will treat you, they call checkpoint inhibitors. Google that, what comes, right? Drugs, checkpoint inhibitors.
Immunotherapy that works to block the protein cancer cells used to hide from the immune system, thereby releasing the brakes on the immune response on the T cells can attack the cancer.
Correct, right? So these are the brakes that the cancer apply to inhibit T cell. We want to block those brakes so that the T cells kill cancers. These are the 2 drugs available. Now if you pay attention for over here as well, like for example, what we can do is that this is another idea. What we do is that we excise the tumor and we look at all the cells which are the tumor, right? And want to see all the clones which are there, right? And find out which is the one that the antigens of this tumor can be recognized by what type of T cells, right? So we find out, take the tumor out, look at the antigens, so the different antigens, right? Grow them, right? Take your T cells from the body and see which one kills them, and whatever kills them, we expand them outside and give it to you. This is called adoptive transfer. Adoptive transfer. Kind of CAR T cell therapy. Right? We make the job of your body easy. Instead of your body looking at all these different antigens and getting confused and generating tons of T cells, we do this job for them. Isolate them, treat them, prepare them, and then find out which are there, grow them in large quantities, put them back. This is called reinfusion of cell. This is not that simple, but I'm giving you the basic idea. So this is also a treatment, CAR T cell therapy, and these are the checkpoint inhibitors for anti-cancer therapy, and this is the immunological function that we have. Okay. You guys have to go at 12 somewhere? Embryonic cells in the womb of the mother, right? So what happens is that these cells can develop into 3 different types. So we can have the cells which are there at birth, or they can differentiate into different types of cells, or they can get the clonal amplification that you call. And then these are the type of antigens that we got. One of the antigens we call oncofetal antigen that you get at birth, and these differentiation pretty much are normal intracellular enzyme, and then you can see some of them are cancer proteins, and some of them are immunoglobulins, right? When the cells which are growing, right, all of a sudden, you remember when the cell go to mitosis, there's a chance of maybe 1 in billion or 1 in 10,000 for them to kind of differentiate into these cells. So this kind of mutant, we call them mutant cellular product. So there are two types of products that we usually have. One is a point mutation. So if there is a sequence of amino acids, there's only one mutation that happens in that gene product, and you can have many different type of cancers because of that. You can see several cancers are there. And then again, there is a P53 gene which actually controls that, which is P53 gene. We call it housekeeping gene. If there is a mistake in the cells, it makes sure that doesn't happen. But if it fails, it gene itself becomes mutant, then you can have tons of cancers. So this is what the what we call pathogenesis of cancer is. Okay? And then the other thing is that sometimes you get viral infection, and viral infection, like human papilloma virus, right? It transforms your gene, right? And then especially some of the proteins are and that's true for cervical cancer. That's why the chances of people who have HPV, right, is higher, because HPV is a human papilloma virus that is oncogenic virus, like even hepatitis. Hepatitis B virus is also oncogenic virus, right? AIDS virus is also. Some of the viruses go and hit your gene. They transform the gene, right? And notorious is cervical, right? Then there is another mutant which leads to. So you can see that these type of cancers that you have, they have been related to a very specific antigen. They found out, just by that method, what are the antigen. The good thing is that once we know the antigen, we can develop a diagnostic test. Right? For example, we have this PSA test. This is the PSA test. We know the test, so we look at the PSA and find out if the person has predisposition to prostate cancer. Right? If, for example, we're going to test your gene for HER2 or new gene, there is chances that you may either have a breast or ovary. So sometimes it runs genetically. This is what the genetic tests are. Not necessarily if you have a gene, you will have disease, but chances are high. Expression of the gene and dominance of gene are two different things. But this is what they do. If you have a Lewis gene, you have high chance of lymphoma. So what we normally do is, if there is a family history of a particular cancer, right, and you do the genetic test and you happen to have that particular gene, then there will be a question mark there. Okay? Especially for these oncogene CEA is done because it has lung, pancreas, breast, colon, stomach. Oh my goodness. That, and then if you have AFP gene, so these are two major genes that we can test you because they got tons of cancers. Right? So these are these tests that you normally do. Some people do, especially the physician would do only when you have a predisposition or family history of that. I don't remember, there was some Hollywood act. I used to teach that maybe 10, 15 years.
Angelina Jolie.
Yeah,
Cancer.
so at that time I was teaching so she probably firstly found out that she is positive for both. So she first went for the breast surgery, an ovary surgery afterwards. But again, the question was that, question will remain ethically that she showed the gene, not necessarily the disease, right? So that wasn't a good ethical example, that if you express a gene, you should get, mastectomy or ovarectomy, because not everybody can afford that. And then secondly, it's going to create a scare among the people, because not everybody has a Hollywood star that he can do reconstruction surgery afterwards. So that was good and bad. So there was a lot of discussion at that time. But again, high level, so these are the tests that normally are available, right? And they keep on looking at so they identify the antigen, and they identify the gene as well. And then again, if in the future a gene therapy comes, then probably we will kind of, look into that. But they're mostly like diagnostic, but you can see types of cancer, and they're usually, this is a very common questions, very common question asked clinically from this slide, because this is something new. I'll just like a minute or two so that she doesn't have to draw it again. Okay, and then again, you can see from here, when they started with this, the first CAR T cell therapy, it keeps on improving. So they had first generation where there's one signaling pathway domain was involved. In this one, you can see. When they improved that second generation, they have two. So you can see a co-stimulatory molecule also came on top of that, right? And then they had third generation where maybe 4 signaling domain will be added. So keep in mind that just finding one is not enough, because it's just like water. The body does water. If you block the water, it's going to find the way, right? So if you look at the signaling pathway, right, the signaling pathway can occur through only this receptor, but if you block that, it's going to go for the other co-stimulation. You block that, it's going to go for different one. So the thing is that they can look at this gene over here, 1, then they have 2, 1, 2, and then they can add 1, 2, 3, 4. So keep that in mind, that we are discovering these gene therapies and drugs based upon the genes. So the future drugs for the cancer again will be looking at your genes, looking at your antigens, looking at the T cells. So this is like the new world of therapy for autoimmune disease, for diabetes, for cancer. So you can see that even the simple experiment that was shown over there, you take the T cell and then you develop a clone, but the T cell again, when you use this T cell, over here, so they're going to use a specific receptor. This is a very specific receptor that you can see from here. So they keep on improving, because the chances are that the more signaling pathways which are involved, the better it is. So there's always a room for development. There's always a room for improvement. So you can see that it's just like only the basic 101 level of cancer immunotherapy. And we are discovering new cells. I just told you last week Tregs. Now the question will be now that drugs, Tregs, or gene of Tregs have already been found in autoimmune diseases. So it's a big field. If somebody were to go to R&D, cancer, they hopefully for good, because they're improving, right? But again, there always are challenges, but they have to understand. Checkpoint inhibitors over there on the right-hand side, they're available in the market, right? CAR T cell therapy is available in the market. The diagnostic tests which are there based upon the antigens are available in the market. Gene testing for the cancer are available. They're all immunological tests. They're all immunological tests. And I even haven't started talking about the antibody. That's the most complex, antibody engineered drugs, because that's the specificity that you'll have for anti-cancer therapy. Any questions? So we'll continue that tomorrow.